I believe this is the most difficult talk of the day, the most difficult topic of the day, because after all these years, I remember Bob with Judah Folkman in the front line of YASCO meeting some 10 years ago when the first positive randomized clinical trial of anti-VEGF was presented. After all these years, we should say first that anti-angiogenic therapy is still an anti-VEGF therapy, and that it's a targeted therapy without a clinically useful measurable target. So this is the big issue here, and I hope I will be able to convince you that this is the area where we should invest more. Why? Because there are at least 50, if not more, shades of complexity in the field that make VEGF the only molecule which is actually targeted, but probably not the only one that should be targeted, not the only one that should be measured, and we should definitely improve. Otherwise, we are really at risk of not being clinically successful for the next years. So the levels of complexity are such that after a long talk from Elisabetta Deiana, I have to add something more. Which are the molecular mechanism for angiogenesis? I mean, physiological angiogenesis, the selection of the tip cells, the stalk elongation, then the resolution of the creation new vessels. All these different complex processes are regulated by many molecules in addition to VEGF. Then you know there are at least 4 ways to vascularize a tumor. We have been talking a lot about vessel cooption. We know about sprouting, we know about progenitors that can be mobilized to generate new vessels, and there is still another neglected way to vascularize a tumor, which is intussusception. Maybe in the next year we will talk more about that. Then, tumor vessels are structurally and functionally abnormal. It has been told many ways, but that's crucial. They are leaky, heterogeneous, tortuous, and so on, which will led to the concept that I will discuss of vessel normalization, we'll talk about later. And then, least but not certainly less important, vessels might be completely different in primary versus secondary cancers. This is again the Francesco Pezzella dogma, which has been mentioned before by Andy Reynolds, and I believe is very, very important. So we should use different tools when dealing with primary cancers or metastatic disease and so on. This is the Francesco's message. I really believe it has been not well covered in all these years. So, in spite of all this complexity, in spite of this all, I still believe that angiogenic therapy, anti-angiogenic therapy, is one of the most successful in cancer therapy, at least in the last 10 years or so, because I don't remember another class of drugs with so many applications in so many different flavors or types of cancer and so on. This is the never-ending list. You see that virtually all, however, is anti-VEGF, apart from the new kids on the block, which is Ang targeting in ovary cancer. So in spite of this, if you look at all the data, you see a common paradigm. There is a benefit, a clinical benefit, frequently related to progression-free survival, not to overall survival of some months, and then patient relapse and so on. But let's imagine together what will be the results of drugs like Herceptin or crizotinib without HER2 detection, measurement, selection of a patient, without ALK rearrangement detection, and the administration of crizotinib only to patients with ALK rearrangement. So probably we would not have seen such a survival advantage. So after all, the fact that we are still giving these anti-VEGF drugs to patients without a selection is detrimental for the results of a clinical trial, but is, on the other side of the coin, amazing for the fact that it's simply working without a patient selection. And what is changing is for the drug company, the fact that using targeted therapies without a clear-cut measurable target is costing a lot. You remember that some 20 years ago, 5FU was approved for colorectal cancer after 200 patients treated because of a large benefit in overall survival. Now we need several thousand of patients because the benefit in overall survival is extremely low, and we need more and more patients. So this is a sort of a drama for a drug company, and this is why the cost of these drugs is skyrocketing. And the target, after all, we should remember, is to pick up out of 100% of the patients the many or few patients for whom the drug is not toxic and beneficial, or the patient for whom the drug is toxic but beneficial if toxicity is acceptable. And we are dealing with a drug that is probably not that toxic to be afraid of picking up also this category of the patients. Coming back to the pharma dilemma, one should keep in mind that biomarkers should be used intensively also at the preclinical level. We're dealing with mice. Because otherwise, if you're not able to pick up and to kill the drugs that are not able to work in randomized clinical trial, and this is actually, Bob told me yesterday, about 60% of the cases in randomized clinical trials, this will increase tremendously the cost of this drug. So it's not biomarkers is not an issue only related to patients. It's also an issue related to preclinical models. And I will just spend one slide about this, mentioning this terrific review by Bob and John Ebos, showing that there is a total imbalance between the preclinical setting models used and the clinical setting model used. So we should use the right preclinical model to investigate whether a drug is going to work in the right clinical setting, which is totally different for primary tumors, early stage macrometastasis, and or late stage metastasis. So please read this review by Bob because I think it should be kept in mind every time we talk about biomarkers. So finally, I was really enlightened some 6 years ago by this review by Rakesh Jain that divided the biomarkers issue in 3 different levels: baseline, dynamic, and escape. I still believe it's the right way to proceed, but some 6 years ago, the forecasts by Rakesh were probably a little bit naive. So my proposal today is to go through this proposal of Rakesh of 6 years ago, to revise it using the same paradigm, divide the biomarkers in baseline, dynamic, and escape, and to see what we can define as promising or maybe is even close to validation in these 3 fields. Let's start with baseline biomarkers. The biomarkers that are measured at time 0 before starting with the therapy and might have predictive or prognostic potential. What are the best candidates? Germline SNPs. We know that VEGF is regulated in a very complex and subtle way by many different receptors, and there are a lot of germline single nucleotide polymorphism for these receptors and even for VEGF. So over the time, there are many different clinical trials that have accumulated data showing that some, unfortunately, rare SNPs genotypes have different clinical outcomes. This is, for instance, a breast cancer study, and showing that this polymorphism had a totally different clinical outcome. Unfortunately, it was not very frequent among patients. So the different polymorphisms indicated are many, not only at the VEGF levels, also at the level of endostatin or at the level of receptors. The problem if you make a meta-analysis of all these polymorphisms, SNPs, if you like the word, you reach a significance that is very, very close to 0, because there are too many of them, actually. So this is at low resolution. I will keep it at very low resolution, the never-ending list of SNPs investigated, and the frustrating results showing that sometimes you have a SNP which is predicted, but it's very hard to replicate in other clinical trials and so on. So we should focus on SNPs that are really clinically relevant. And to the best of my knowledge, this is probably the most convincing one. It's a SNP at the level of VEGF receptor 1, investigated by the Belgium group in a randomized clinical trials in pancreatic cancer. You see that the addition of bevacizumab to gemcitabine and erlotinib really had no effect. But if you just pick the patient with different SNPs in this VEGF receptor 1 gene, you see that there is a clear-cut and dramatic stratification of the outcome according to the genotype, which was not this is for patient receiving bevacizumab, this is for patient not receiving bevacizumab. So this is clearly bevacizumab related. And why this SNP is so important? Because it's very clinically relevant, because it's located in the tyrosine kinase domain of VEGF receptor 1 gene. So to me, this is albeit not being published yet, the only strong evidence that a SNP can be really predictive, at least in pancreatic cancer, at least in patients receiving chemo plus Bev. So there is a hope at the end of a tunnel, of a SNP tunnel, if you like so. Okay, other possible baseline markers, immunohistochemistry, again, very frustrating results. Probably the only consistent results is that in some hands, immunohistochemistry CD31, at least in colorectal cancer, can be predictive. You see some difference in this patient with very high expression. Neuropilin in some other colorectal cancer studies, and this is all I can propose you so far. It's frustrating, but this is the reality. VEGF levels, of course, this would have been the dream of all the investigators in the field, because just measuring VEGF is pretty simple. Unfortunately, for many years, we have used an ELISA test, which was not very sensitive, at least at the bottom and top of the spectrum. Then Roche, which makes bevacizumab, came out with a new generation ELISA that were used for retesting all the randomized trials. And this new generation testing was really successful in picking up by baseline levels the patients, for instance, again, in pancreatic cancer that were supposed to receive a benefit. This is the pancreatic cancer. You see that compared to old-style VEGF levels measured with the old ELISA, now the curves are more open, and there is a significant difference between the levels of plasmatic VEGF. This is the AVITA trial, again using this novel ELISA. Taking all together these results, this is the Avado for breast cancer. So you see that using this novel ELISA, which unfortunately is still not available, this is the real pity, at least in the hands of Roche investigator studying retrospectively the plasma of their randomized study, you see that this approach has predictive potential, sometimes also for overall survival in several trials that were simply negative by not using this approach. I've been told that now Roche is trying to enroll patients according to this stratification. I really hope if this is true, and I really hope to be able to give you good news in a couple of years about this. Another approach may be to combine VEGF along, for instance, with neuropilin. You see that, for instance, in gastric cancer, combining VEGF with neuropilin can give you some stratification of the patients. It's probably not a lot convincing, but this is the best we can do now. Or another approach, for instance, in hepatocellular carcinoma, is to combine VEGF with a plasma insulin-like growth factor. You see that the curves are opening a little bit if you use together these two levels. Again, the problem is that it's likely that these patients are very few, so maybe it's not clinically that useful. So this is the list of the data revised some 5 years ago by Adrian Harris and Adrian Jubb. And again, it's frustrating because there are different approaches in different clinical trials, and nothing can be really defined as clinically validated. Then the baseline potential of cells with an endothelial phenotype, monocytes, and so on. There are a lot of work in this field. We started along with Bob Yuval some 10 years ago in investigating this field because there are so many different cells that are possibly involved in the vascularization of the tumor. So we realized again with Bob, Yuval, John Amack, a very complex flow cytometry approach that at least in some specialized laboratory gave some very interesting results. And in mice, this approach was able to predict the optimal biologic dose of different antiogenic drugs. So we moved to the clinic, and not only in Milan, not only in Paris, but for instance in Nottingham, in the UK, this approach was able to stratify the patients according to their risk. This is the fantastic parallel between the circulating endothelial cells measured in the UK and the Nottingham Prognostic Index in breast cancer. And actually in breast cancer, this approach had some stratification predictive potential. I'm showing here quickly some 3 published clinical trials in Milan. This is metronomic chemotherapy. This is metronomic chemotherapy plus bevacizumab. This is again metronomic chemotherapy plus bevacizumab or erlotinib or herceptin. Again, you see the stratification curve is very open, and there is some good predictive potential. There is predictive potential also in other diseases like glioblastoma. This is a study we have done and published last year with some neuro-oncology surgeons and oncologists in glioblastoma against using with flow cytometry approach. This is a Japanese group, again, finding some predictive potential. Overall, I cut this as 2012. There were, up to 2004 and 2012, there were some 30 studies demonstrating some clinical potential. In 2015, it's getting closer to 50, but you see this curve. This is the number of published papers on the field. And you see there is a decline after 2011. We reach a number of about 50, but there is a decline. Why? Because the procedure is very complex. You really need a dedicated laboratory, you need fresh blood. So this sort of approach can be done in specialized laboratories, but not in all the laboratories. And I really believe that this approach has a lot of potential, but it will not be reproducible in the best way unless we have a cheap or very, very simple, very reproducible approach on which we are still working. Okay, what is a dynamic biomarker? It's very important, but very simple. Is answering to the question, I started the treatment, but is it really working? Of course, the simplest way is to do some imaging, and there was a splendid talk about that. I just would like to remember a paper that was published some 6 years ago in the JAMA, and maybe it was not adequately studied by people in the clinical field because it was on JAMA and not on the Journal of Clinical Oncology or Lancet and so on, showing that the RECIST criteria, and it was described today, simply does not work for anti-angiogenic therapy. One should use the morphology criteria. So the simple measurement of the diameter of a lesion does not work for anti-angiogenic therapies. This is colorectal cancer receiving bevacizumab plus chemo. And you know in what a dramatic way the Q survival curves, this is overall survivors, are opening just by using morphology response criteria versus the RECIST. And this was true not only for the surgical cohort, but in some way also for the patient with unresectable tumors. So the take-home message, but very important message for the clinicians, is never trust the RECIST criteria more than a morphology criteria. Talk with your imaging people. Maybe we can discuss it later. And I've been really surprised over the year that this approach has not been considered for as a standard for imaging evaluation of anti-angiogenic therapies. Blood pressure. Blood pressure has been proposed and validated in some setting as a dynamic biomarker because of data like this, showing that an increase in the blood pressure of a patient was associated with a better overall survival. This is, for instance, lung cancer patient treated with bevacizumab in combination with carboplatin and paclitaxel. This is a very simple approach. It's very reproducible. It has been seen also in some way in pancreatic cancer patients, even though this curve, you see, became a fish and not a trumpet after some months. What is intriguing for me in this blood pressure hypothesis is that it is related to some other measurable effects like the amount of nitric oxide, prostacyclin, and so on, but most of all is related to systemic pruning, which brings us to the concept of how anti-VEGF is working. Is working by pruning vessels or by normalization of the vessel. If it works by pruning, then blood pressure is really a relevant marker because pruning is supposed to increase the blood pressure. If it works by vascular normalization, then most likely you should see another effect and no effect at all on the blood pressure. You know that the vessel normalization hypothesis has been studied by Rakesh and Peter Carmelit for several years, and they came out with a concept that vascular normalization is the only mechanism of anti-VEGF therapy because it can reprogram the tumor microenvironment to a less hypoxic environment, so not stimulating cancer stem cells, maybe having an effect of immunosuppressive action of the environment and so on. And they also were able to generate some data, for instance, in lung cancer, telling that when you have a normalization, of the vessels measured by imaging, then you have a better survival. On the other hand, there are data published, for instance, by this Dutch group and by other groups, suggesting and showing a totally different approach, a totally different results, in the sense that after bevacizumab, the distribution, for instance, of radiolabeled paclitaxel or other drugs was reduced. So this is not compatible with a normalization effect and suggests actually that there is no normalization. So I do believe that at this time the jury is out. There is no formal proof of a normalization effect for all the types of tumors. I believe the best we can say is that in some patients, in some types of cancer, there might be a normalization associated with an overall survival because of more oxygenation and so on. In some other types of cancer, it is the opposite. This is my take-home message up to now. But this is still a fascinating field, and there is a lot of further work to be done. Another dynamic biomarker that can be of interest, it's strange that it has not been more investigated over the years, is a simple measurement of plasma VEGF after a couple of months of therapy. For instance, in this cohort of breast cancer patients that were treated for, in some cases, for more than 1 year with metronomic chemo plus BEV, you see that VEGF after 2 months was the better predictor of survival. And if you reach a level of VEGF inhibition up to this plateau, you really had a better survival, and patients were treated in some cases for 1 more year, despite the fact that they were metastatic. So I believe there is some potential also for studying better the circulating levels of VEGF as a dynamic biomarker. And the last biomarker field is the escape, which is fascinating because there are several different hypotheses that are summarized here of how a tumor can escape from VEGF therapy. And what we have seen in many cases in patients receiving metronomic chemotherapy and anti-angiogenic therapy was that, for instance, the number of circulating endothelial cells is dramatically reduced at progression, which is the opposite that one can expect. You see, you escape from an anti-angiogenic therapy with more angiogenesis, but this is not the case. You escape from an anti-angiogenic therapy by reducing the number of cells within an endothelial phenotype. So what is happening? To make a very long story short, what in the laboratory we have seen over the years was that the pressure of anti-VEGF therapy generates these super vessels, which are very large, tortuous, dysplastic, and very frequently infiltrated by immune cells, which is very, very interesting. So how can you set up a new line of therapy after this super vessels is generated? What is the best next option for these patients after maybe several years of anti-VEGF therapy? Maybe that an approach which inhibits a crossroad of different molecular mechanisms is the best approach. We've been working for several years for the inhibition of ALK, which is at the crossroads of different angiogenic pathways related to TGF-beta, BMP9, endoglin, and so on. And the results were really encouraging in the sense that the addition of an anti-ALK1 antibody was able, at least in mice, to revert the resistance to anti-VEGF, and this approach was extremely effective against super vessels that were inhibited by the addition of an anti-ALK1 antibody. And I can tell you there are some new data coming out about this field that are suggesting that this anti-ALK1 approach can really work in patients receiving anti-angiogenic therapy. But vessels are not simply tube, and it's becoming more and more clear that targeting the immune system in addition to the tumor cells, in addition to the vessels, is potentially a sort of home run in cancer therapy. You are all aware of the very interesting results of inhibition of the checkpoint in cancer cells. And this is related to angiogenesis because all the vessels are in some way infiltrated and many different immune cells are present. And I believe that maybe the most interesting new data in the field are generated after Napo-Ferrara showed that refractoriness to anti-VEGF is determined by the ability of tumors to prime and recruit some immune cells. He was focusing on myeloid cells, but now there is evidence that also lymphoid cells are very relevant. For instance, this is a paper of 1 year ago showing that the simultaneous blockade of PD-1 and VEGF-R2 in this sense, can induce a synergistic and very strong anti-tumor effect in vivo. And this is related also to the biomarker field, in the sense that there are some new data showing that the association of PD-1 measuring tumors and VEGF measuring the plasma has a lot of predictive potential in patients, for instance, with kidney cancer treated with TKIs against VEGF receptors. So I'm astonishingly almost on time, and I'd like to summarize my take-home message by telling you that we just have candidates. We are candidates for biomarker potential that are getting stronger and stronger every day, but are not yet validated for a series of reasons that I try to summarize in my slides. And so far, the best predictive might be SNPs, when you are able to pick up a SNP with a biological relevance. Cells, this new plasma circulating ELISA test is highly promising, but we have to, in some way, to wait for the commercialization from Roche, and it would have been really a pity if this new ELISA test would not be available for all the clinical trials. And neuropilin maybe is an interesting potential biomarkers. For the dynamic biomarkers, I believe that measuring hypertension has a lot of potential, but should be used also to discriminate for patients where a normalization effect exists and patients where a normalization effect is not seen, or there is vice versa, an effect because of pruning. I talked about imaging and the morphology response criteria potential. Again, I believe we should measure plasma VEGF maybe with a new ELISA test as a potential dynamic biomarkers. And then for the escape, I believe that supervessels and the PD-1, PD-L1 checkpoint inhibitors are really promising and should be studied along with expression of these markers by tumor immune cells as biomarkers. Just let me mention all the people in the lab that really made all this work. I'm just the speaker, but all the data that I've showed were generated by Patrizia Mancuso, Ines, now in Madrid, Stefano Recchioni, Giovanna Talarico, Francesca Reggiani, Cristina Rabascio, Angelica, Chiara Corsini, Giuliana Gregato, Cinzia Massaro, Jessica Quarna, and Pierluigi Antoniotti. And of course, the fantastic, terrific collaboration with Bob and Yuval all over these years. Thank you very much.
You mentioned the ELISA kit from Roche that picks up the short isoforms of VEGF, and the splash that was made a few years ago, I think 3 or 4 years ago now, that based on retrospective analyses of completed phase 3 trials, it looked like it might be a predictive marker. And you pointed out that the details of that test have never been published. My understanding, and this is anecdotal, is that they have been unable to reproduce the data. And that test has been abandoned. I'm pretty sure of that. Now, again, this has been mentioned at a couple of different recent meetings. It's very disappointing. You know, there were people involved like Gordon Jason in the UK who was involved in that test. David Miles, I think, was also involved with it. There are a number of people, but my understanding right now is that it's not being pursued, which would be a real disappointment because, as you pointed out, this looked like something that was logical, you know, that it did go after the target, VEGF, but that's the way it stands right now, as far as I know. Have you heard anything beyond that?
No.
Anyone else? No. Okay. Well, that's your bit of gossip for today. Questions? Anybody?
Andrew.
I might make a comment. I'm really glad you showed the imaging of the liver from that JAMA paper, because I think that distinction of using novel imaging criteria to measure response is really important. I've spent a lot of time talking to Evan Lawyer about why that hasn't been adopted, and the problem is that it's to do with the quality of the imaging. So in Evelyn Lawyer's CT scans at MD Anderson, they use very high radiation dose, which gives beautiful images where you can really measure those changes. But the trouble is, when she came to Europe to look at CT scans, they use lower radiation dose, and the border, and I don't know if you would follow this, of the tumours was less well defined, and it was more difficult to apply the marker. So I think it's a technical thing with imaging between different centres that's difficult to, so that needs to be standardised. Imaging biomarkers need some standardisation. So this is more of a comment than, based on our experience. But then also, I think there is a paper from a Japanese group that have validated those criteria. So I think...
I totally agree. I really had no time for entering into this technical detail, which I completely agree. And it's just a matter of having a very good relationship with your radiologist,
yes,
I believe.
Absolutely.
And telling them that you really, for this type of treatment, you really need a different approach. Because otherwise...
No, I'm not going to mention MR, actually. My question really is, so the radiologists in general are very happy to interpret images in whichever way that's suitable. I find that it's pharmaceutical companies who don't want to adopt new techniques. Do you have any comments on that and how we would get pharmaceutical companies to accept new imaging?
No, you're totally right, and I just have a very broad and sad comment, which again can in some way be added to Bob's consideration about the fact that Roche is not delivering any specification about why their ELISA new kit is not available. The agenda of pharma is clearly towards giving the same drug to all the patients. So it's conflicting with the agenda of precision medicine. Despite the fact that over the years there is an evidence that for a precision medicine approach, pharma can receive a huge benefit by using a drug for the lifetime of a patient. I remember by the time Glivec was in preclinical development, the former Sandoz, Cibat-Geigy was not interested because they say it will not work. There are not so many chronic myeloid leukemia patients. So why should we work on this tyrosine kinase approach? After all, this drug is a blockbuster, selling $7 billion every year just in Europe or in the States, and is likely to be given to the patient for their lifetime. So we should discuss with pharma the fact that in precision medicine approach, maybe that a drug will be given not to all the patients, just to a fraction of them, but likely for their lifetime. And this is the only argument we can have with them.
You know, maybe I'll before we move on, I'll mention one thing that's I think is quite interesting about, maybe it's a combination of like a predictive and pharmacodynamic marker, and it also relates to, I guess I wouldn't call it precision medicine, individualized therapy. So this is a trial that's being run now in Canada. It's in renal cell carcinoma patients. It's based on the work of a renal cell oncologist named Georg Bjarnason. He actually works at the same center, same hospital as I do. So let me take you through it very quickly. Maybe it could evoke some interesting responses from the audience. What he has, Dr. Bjarnason has reasoned for a number of years now, at least in his patients, kidney cancer patients, mainly clear cell renal cell carcinoma, say patients getting sunitinib as first-line therapy, is that roughly 25 to 30% of the patients are probably not responding because they're getting a sub-threshold dose of the drug. And this is based on toxicity. So what he's noticed is that, you know, a number of patients, and these are often patients who don't seem to benefit initially from the therapy, don't have overt evidence of, say, hypertension or some other toxicity marker associated with giving the drug at the dose that's recommended. So that's the flat dose of 50 milligrams every day for a month and then the 2-week break. So what he's been doing, he published a retrospective analysis of this idea of adjusting the dose individually over time.
Up to toxicity, you mean? So
It-
you are back to maximum tolerable toxicity.
Yeah, in this case it is, because what he says is, let's say he starts with the 50 milligram dose. If patients don't show evidence of a certain level of toxicity, then he adjusts the dose upwards to 60. If they still don't show evidence of a certain toxicity, then he'll adjust the dose upwards again to 75, and not only adjust the dose, but the schedule. He's always felt that the 2-week break was too long a period, the 4 weeks on, 2 weeks off. So it's more like 1 week on, 1 week off, or 2 weeks on, 1 week off. But you're adjusting also the schedule individually over time, and also the dose. So the retrospective study analysis results were published in 2014 in Urologic Oncology, I think. And based on that, Pfizer has initiated a prospective trial across Canada, a multi-center trial. So I think this is really also quite interesting, because again, it's sort of a combination of predictive and pharmacodynamic, and it relates to the issue of individualized therapy.
Yeah, that's fascinating. And if I just have time, I have another comment along this line, is that tyrosine kinase inhibitors are very dirty drugs.
Mm-hmm.
We are focusing on the 3 kinase that we know better, VEGF receptor 1, BCR-ABL, but there are 20, 30, 50 more tyrosine kinase inhibited, which we really don't know a lot about. And this will lead, in my opinion, in the future to a lot of reposition on this drug. For instance, axitinib now is considered the best inhibitor for an isoform, a particular form of BCR-ABL. But this went out after 5 years of clinical use and by serendipity. So we really know not enough about tyrosine kinase inhibitors. For instance, ibrutinib was supposed to work only by targeting Bruton tyrosine kinase, but it might be that it targets also some kinase involved in immunosurveillance of cancer. So it's a field that should be reconsidered in the future, considering all the kinase that are still neglected so far.